Okay, so hello. Well, this is an experimental stream that, well, I will be looking at genomics papers, and I thought, well, might as well stream it a little bit and try out also this medium of the YouTube and other similar websites. As you might feel, this is a new territory for me, so it's well, somewhat of a new stuff, and I also need to check if my microphone is in, because this is not normally. Hello? Desktop? No. Microphone. Are you on? Oh, there are people in the chat. Hello. Do you hear me? If somebody can make a chat message. Okay, I'll try again. Is the sound on? Sent. Excellent. Thank you, Sabine. Good. Oops. That's. I did something. Sorry. Oh, go back. No, the problem is that I don't. I would like to see simultaneously the chat as well, and that is, I'm trying to do that on my phone. Okay, now it's back on my mobile phone, and yeah, it's okay. So that because I just have one monitor. Excellent. So, Sapien, how are you doing? Any tips for nice genomics papers or other? Could you are your own as well? Okay, so my thing was that I seem to remember a paper that was dealing with genomics on what was it? On medieval battlefields, I think in something in the Nordic countries, and so there was basically the thing was that they collected samples from a medieval battlefields, and yeah, just try to analyze that. Could you make a series from raw data retrieval to analyze a single cell RNA-seq? Unfortunately not. The raw data stuff, probably yes, depending on what kind, but the generally the sequencing is not really my territory, and not the single cell RNA-seq, not at all. And honestly speaking, I try to avoid topics that I don't understand fully, because then it's might as well you're getting a wrong advice. So that's why what I want to, or try to focus on things that I understand very well, and therefore I'm confident that whatever I tell, it's valid. And for the RNA-seq, it would not be the case. So unfortunately, that's a negative. But well, I'm happy you're here. So if there is anything else I could Uh, the, what is an SRA? Then it also kind of shows that I'm not very SRA. I don't know. Actually, I'm on Google, so. So I don't know if this would be this thing would be public yet, but SRA toolkit. Oh, this is also something with the thing. Oh, okay, that might be so to retrieve ASCII data from geodata set. Well, that I don't know either, but well, I just make a note. Actually, I make a note, and this is seems more plausible. Still, I need to find out what it is and how, but SRA toolkit. Now the problem is that I have far too many things on the plate. So I also, if you might notice, there is from the videos there is a bit more kind of well, not so frequent as of late, and that's because there is a bunch of other stuff I need to deal with. So well, anyway, so thanks for the tip, and the best I can say that I will see what I can do. Don't expect anything immediately, I would say. Okay, but now this papers on the okay, so let's go full simple here. Medieval genomics paper. That was something with the Viking, I think. Vikings. Population genomics of the Viking world. Not what I wanted, I think, but still. We like population genomics, so not found. Ah, come on, nature. Okay, all 41 versions. This seems to be more. Okay, so again, this is not found. Europe PMC. No, okay, now this is, now we are talking. Freebase, no, also. Hello, Himani. Freebase rings a bell, honestly, but I can tell you for sure that I did not use it. Again, so this is variant calling. This is also then, yeah, of course, yeah, that it goes in towards the sequence analysis, which I do surprisingly little about. Well, I have a kind of a basic understanding, and I think I did once or twice, but it's not something I would do routinely. So I had a PhD student who I was lucky enough to do all this for me, and actually he did a very good job on dealing with sequence data. But so I'm more of a kind of not as experienced in this thing. Okay. So Greenland, global influence expansion, find Viking periods involved in gene flow into Scandinavia from south and east. We observe genetic structure within Scandinavia with diversity hotspots south and restricted gene flow within Scandinavia. We find evidence of major influx of Danish ancestry into England. Well, that's I'm wondering why is this the case. Of course, not wondering that much. Swedish influx into Baltic. Oh, Baltic, sorry, I was on this. Okay, that's fine. Norwegian influx into Ireland, Iceland and Greenland. Additionally, we substantial ancestry from elsewhere in Europe entering Scandinavia during the Viking Age. Ancient DNA analysis also revealed that the Viking expedition included close family members. Any of you have seen the movie, was it Vikings? Was it? Yes, this one. This guy. It's historically not so accurate. Ragnar Lodbrok. Good series. Historically, as I said, historically not as accurate, but entertaining for sure. Anyway. And there is so. Conclude that the Viking diaspora had characterized a substantial trans-regional engagement. Yeah, this is a nice, very nice, nicely written sentence here that the characterized a trans-regional engagement. In other words, war and conquest. Distinct populations influence the genomic makeup of different regions of Europe. Also negative selection, I guess. Artificial selection via X and other tools. And Scandinavia experienced increased contact with the rest of the continent. This is a beautiful sentence. Putting the history into a genomic context, which is beautiful, just beautiful. Full text. I definitely don't want to read the whole paper as of now, but would be nice to see their. Oh, now it's not, the nature thing is also working. Would be nice to see their figures, what they have. Okay, so what is this? Bronze Age, Iron Age, Early Viking Age. Oh, there is a lot of things going on here. And medieval and early modern. Okay, so let's see. Early on, well, everybody was a bit active. Early Viking Age Swedish were surprisingly active, or at least, and a bit of Denmark, nothing Norway. Okay. And then whatever the Viking Age is, there is everybody was doing their share of what they told trans-regional engagement. Yes, so they engaged trans-regionally. Just too funny. Okay, what is this? Multidimensional scaling of, where is it? Multidimensional scaling of quite a lot of ancient genomes on the basis of pairwise identity by state sharing matrix. Identity by state, huh. Interesting. That is, no, I'm just wondering because the identity by state is normally very similar across or more similar across populations, I would guess. But other ancient samples. Outlier individuals with the hunter-gatherers, Sami-related ancestry. Nice. No? That's quite nice. Okay, folks, any other kind of recommendations for some interesting papers? Yeah, I did this one. Basically I came with an intention to look at this paper first, but then I am a bit of a Yeah, and the other stuff what I wanted is a bit of a, yeah, just really try out the streaming stuff without camera, I would say, because the big advantage of not having a camera is that you don't have to look nice. And it's like normally I sport the shirt type of like appearance, but well, not now. Okay, so long-term genomics. I'm a Viking. Am I? That's a question. New Genetics and Society by Strand and Colleen. DNA, popular culture, and construction of geneticized, is it how it's geneticized identity? I guess this will be in the direction of the like all these DNA tests, and they are with the Viking proportion. In this article, we know how genetic genealogy reshapes popular notions of historical identity, as it facilitates a genetically informed understanding of ethnicity and ancestry, drawing on interviews with Swedish, British, and American individuals who have employed genetic ancestry tests, I guess, GATs, to prove ancestral connections to Vikings. We explore how the desire to be a Viking, well, it's not a bad desire, is articulated through a convergence of pre-existing disclosures around Vikings and DNA. What? That's a bit complicated sentence, but anyway, so they want to be a Viking. That's the kind of the gist of the sentence, I guess. By combining science from genetic science and popular description of Vikings, our interviewees create a new discourse of geneticized Viking identity. Well, it's a strange paper. In this new discourse, socio-historically constructed ideas about Vikings are naturalized as the innate qualities of individuals who possess certain genetic possess a certain genetic composition. By how? Images of the Viking, once created for political, cultural, commercial purposes, revived in a new embodied form, so you can start circulate a new social context. The association appear to be confirmed by genetical science. What? Is this a genetics paper? Or what? New Genetics and Society. What is this? Oh, okay. It's too much of a socio-historical things here and too little about how to say genomics in this at least in this abstract. So honestly speaking, okay, materials and methods. This anime is from a multidisciplinary research project investigating meaning-making practices of around historic DNA. What? Well, it seems to be a bit more complicated issue. And this is revolving around the genetic ancestry texts. Unfortunately, not so many figures that we could check on. And a lot of text that I'm guessing is around the same, maybe conclusions. Oh. Berserker perception. Okay, I'm just, well, taken out of context, but, okay. Vikings as entrepreneur. Oh, well, this is not, this is not, not a genomics paper, right? This is clear from here. Still a long-winded conclusion. Okay. In this article, we have explored the desire to be a Viking among the group of individuals to claim Viking ancestry. Come on, no. Sorry, no, this is more like a, well, storytelling type of, no, this is something interesting title, but absolutely storytelling type of situation. The battle for common ground. What? Okay. Oh, because I had the battle in the title. Divided by the uneasy relationship between archaeology and ancient genomics. Two fields in the midst of technological revolution are struggling to reconcile their views of the past. Okay. Meaning what? But this is ancient genomes. Oh, wait. More than 1,300 genome sequences have been produced from the remains of ancient humans, sometimes challenging the historical narratives derived from artifacts. Well, yes, I guess new proofs or methods become available to explore the past, and then our understanding of the past is changing. That's fine. Europe, North America, South. Oh, this is the thing. How many? But what are these dots? These are studies of what? Sequences probably. And this is from there where the largest ancient DNA study to date, scientists sequenced the remains of 400 Neolithic, Copper Age and Bronze Age Europeans, including 226 associated with the Bell Breaker artifacts. Okay. This is the study. And here it's single dot here. A 4,500-year skeleton from the cave in Ethiopia provided the first ancient human genome sequence from Africa, providing context on movements back from Eurasia. Scientists expect many more examples from Africa this year. Okay, unfortunately I'm not too knowledgeable about archaeogenomics, but it seems that most of the samples are from Europe. Africa and Asia are somewhat represented and the other continents barely. Okay. Well. Okay guys, so like this is an interactive session, so just you might notice there is a chat box nearby, so feel free to use it, or well, I just tell me how you've been, what you are up to, and also if you have any interesting paper you came across, then of course I'm happy to hear about that. Could be also your paper. Also, as you see, I was dealing with the ROH runs of homozygosity parameters at the moment, which will also come at some point also to Genomics Bootcamp. What else now? What else we can do? Following this line of thought. Genomics. And not QQQ, that was just a placeholder. Let's see. Okay. What is this? Okay, horses. Who likes horses? To the right. In Search of the Great Horse, a zooarchaeological assessment of horses from England, AD 300 to 1650. Popular culture presents a deep-rooted perception of medieval war horses as a massive and powerful mounts, but medieval textual and iconographic evidence remains highly debated. Just a kind of a side thing, just came because of the wars. There is a new stuff coming out, which is called Warhorse game. What? Not Gamefall, come on, game. What? Warhorse Studios? No. Okay, then. Oh yeah, this one. Oh, it's Warhorse Studios. Okay, it doesn't matter. Well, anyway, Kingdom Come. Second part will come out. But anyway, that's. Oh, there was also Warhorse type of thing. That was anyway. That's absolutely off topic here. Okay, so the size of the medieval warhorses was highly debated. Furthermore, identifying warhorses in the zooarchaeological record is challenging due to both a paucity of horse remains relative to the other domesticates and the tendency of researchers to focus on osteological size, which makes it difficult to reconstruct in-life usage of horses and activity-related changes. This paper presents the largest zooarchaeological dataset of English horse bones, well, almost 2,000 bones, from 171 unique archaeological sites dating between AD 300 and 1650. Using this dataset alongside a modern comparative sample of known equids, almost 500, we examine trends in size and shape to explore how the skeletal conformation of horses changed through time and reflected their domestic elite and military roles. In addition to evidencing the generally small stature of medieval horses relative to both earlier and later periods, we demonstrate the importance of accurately exploring the shape of skeletal elements to describe the morphological characteristics of domestic animals. Okay. Furthermore, we highlight the need to examine shape variation in the context of changes and biomechanics to address questions of functional morphology and detect possible markers to artificial selection on the past horses. Now this is a beautiful example of the interdisciplinary research. This is like in these last few lines here, there are some very different fields mentioned, and it's kind of a nice example how to combine, really nicely combine fields of science into one. And what we have here is genomics with the artificial selection here. But again, if you walk backwards, morphology. So this is like a morphology of bones and all the just features. Biomechanics. So this is also a separate field of the movement. And then it's also the shape of skeletal elements. Also, this is for the morphology. But and then of course archaeology is there, and history. So really nice example of a how to say interdisciplinary research. So Carly, Amin and colleagues, excellent work, I think. yeah, this is a journal that I don't read so often. Journal of Osteoarchaeology. Okay. I have a feeling that there will be not so much genetics and genomics inside here, but yeah, still there is a, well, we can go through the paper in briefly. Anyway, so there is this what I try to also see here is, or kind of apply, is there is a video on my channel, and also there is this paper on reading papers, and that is how to go through the papers quickly to get the gist what they are about. So that basically is telling that you read the title very well and the abstract, and go through and the subtitles. But well, in this case we don't have much because we have just met the classical ones. But so we're not reading the text, but we are focusing on the tables and figures. Of course, the abstract and then the conclusions. So this is the only things that we are looking at, and well, because it's more like a free roam here, so then we can do it like also as a just check out also some interesting other stuff as well. But data single sample size is period A. And this is where they got them from. Okay, so this is the Britain. I don't know if this is technically Scotland already, but well, anyway. Sites include, due to the geographic proximity, some locations have been combined. Unique sites represented the points as each region. Individual size information and mapping coordinates can be found. Table S1. We are not as interested, but it's nice to have. Okay, results. What is this? Box plot showing wither heights. Okay, this now we're talking in meters. Okay, calculated from greatest length of long bones of archaeological green and modern equids. So these are the archaeological ones, and these are the modern ones here. Okay, displays in wither's height values are displayed in the centimeters and hence high. What is that? On the y-axis and grouped by chronological period. On the x-axis, color figure can be viewed. Okay. So this is quite interesting figure. So let's see. Let's see what we are getting. So withers high in meters. Okay, so later, but oh, okay. Interestingly, everything like the sizes of horses doesn't. So this is the box plot anyway. So the box plots are. So this thick line here, this is the median. It's not the average; it's the median. And the box plot itself. So this is the upper. So this is within the box there is from 25 to 75 percent. 25th to 75th percentile. So this half of the data is within these boxes, and these one, the kind of things, these are, I think, the rest of the so this is the bottom 25, and this is the upper 25th percentile of the data, if I'm not wrong. But anyway, so this is the and so you see that the median length is kind of unchanged throughout the times. In the Norman, Norman times it's kind of went back, but it's like, honestly speaking, we are talking about 10 centimeters here, so it's like, oh no, not even. So this should be like, I don't know, 35, 33, and then down to, yeah, it's about 5 centimeters different, that kind of drop here. And then there is a huge post-medieval, okay, late medieval is still low, and the post-medieval times we have a sizable jump here, at least compared to the other ones. So maybe the large Warhorses kind of things, it's coming from this period, which kind of would make sense a bit because it's like closest to our time. So probably it's. I'm making too many assumptions here, but anyway, I wanted to check out on the or compare to the current times, but this is already the authors did that. So donkey, pony. So okay, so what we see here is that the medieval horses were definitely larger than a pony, but not by much. Honestly speaking, the Percivalski is a it's also like it's a small horse. This is a small horse, and this was about more or less. And it's a mule. Mule is, well, I don't know how big a mule is, but not large. So it was quite small actually. Riding horse. Well, the current riding horse is much taller than the medieval horses. And the warm bloods and the draft horses not even. Well, these are large, honestly speaking. These the warm blood I know, but the draft horses are huge. So anyway, that was an interesting piece of information, although even if it's not genomic. A lot of figures or kind of numbers, width, length, and lengths of these archaeological modern. Okay. And things on the robustness. This is just way too complicated. Once I had a discussion with a colleague of mine who was basically telling the discussions was about this. So my point was that people are on a conferences in general. It's not a problem to show a graph with more than 2 lines, or like I wanted to include 4 lines, and then my colleague was arguing that even if you go to a conference, it's best to show a graph with maximum 2 lines on it, and people best comprehend that what's going on. So I was disagreeing at the time, but seeing this figure here, I go towards this side of the argument, at least in this setting, because. Honestly speaking, you cannot show this one in a conference. Well, this is of course a very different setting here than a conference, but just way too much information here. So plots, means, ratios, measures. Okay, discussion and interesting paper. Anyway, so conclusions. Let's see what they conclude. Despite the tendency of both historians and zooarchaeologists to focus on the overall size of past horses, the results of this analysis suggest that neither size nor limb bone robustity alone are enough to identify warhorses in the archaeological record, as the historical record indicates by remaining more notably silent on the specific criteria which define the warhorse. It is much more likely that throughout the medieval period, at different times, different comparisons of horses were desirable. I cannot read. Conformations of horses were desirable in response to changing battlefield tactics and cultural preferences. The breeding and training of war horses instead was influenced by a combination of biological and cultural factors, as well as individual behavioral characteristics of the horses themselves, such as temperament, which would make a lot of sense. This work has highlighted avenues of further exploration into the biological and functional characteristics of equids used in combat. For instance, detailed examination of the morphological variation of the lower limb bones as well as associated enthesial changes has the potential to decipher these biological trends further and aid in the identification of archaeological warhorses. Furthermore, the incorporation of the ancient DNA analysis presents the possibility of uniting ancestry-related changes as the impact of the introduction of European horse breeds on English stock, whereas advances in ancient genomics allows detection of traits previously unidentifiable from archaeological bone, including coat color, speed, and temperament. Speed and temperament, interesting. Probably because of the various, if they genotype it and then if they find variants that are kind of matching with the today's variants and they know about their functions, that would be my guess. Because otherwise, well, the coat color is clear, I think, because that definitely there is no coat remaining in the archaeological findings. That is for sure decomposed. But for this one, they definitely, if they for the color, there are some genes that are very prevalent. So or kind of, well, we know what genes are kind of causing which color in horses and other species. So, and I think then along the same lines as they can kind of figure out coat color of the horses that are dead for, I don't know, 500 years, then the same thing is, although to a lesser extent, but I think the speed and temperament is the same thing. So if they find genes, or not genes, sorry, variants that could be linked to various characteristics of speed and temperament in today's horses, then I think that is how it can be linked to the old ones, which is actually a very interesting idea in general. And this is also kind of one of the ways how we can use genomics to learn about the past, because I think it is a very nice idea that we genotype or sequence archaeological findings and samples and see what these individuals or animals were, or well, humans as well. But well, I'm more into animal genetics and genomics, but it's of course the same thing it goes for humans. For some traits it works better. For example, coat color here, I think there we can have a very good understanding. Well, depends a bit. Well, it's coat color genetics. It's kind of not, sometimes it's presented as a straightforward thing, but it's not. Honestly speaking, it's not straightforward at all. But I think it's a bit better than speed and temperament being functional, not functional traits, but quantitative traits. So, well, anyway, so finally, going forward, and perhaps most significantly, it's a good place to stop the reading. It's when there is the next thing is the most significantly. But anyway, most significantly, the context of horse remains must be considered for the identification of warhorses. Context, okay. Given the different dispositional processes of horses from other domesticates, as well as the tendency of the horse carcasses to regularly go through post-mortem processing, the search for the great horse must move from castles and battlefields to knackers, yards, and domestic middens. Well, okay. I did not foresee this turn of events here with in the conclusions. Okay, well, perhaps they enlighten us a bit more within the discussion. But yeah, so this is a bit surprising ending here. But well, okay. Good. Do you have the data supporting information? Caption. Anyway. Nice, very nice paper. In search of the great horse. Good, thumbs up. Okay, what else? What else? Ancestry.bomb. Genealogy. Genomics. Mischief, mystery, and southern family stories. This seems like a dangerous paper. No, rather not. Even though it's late, but pandemics past and present role of biological anthropology in pandemic studies. No. Genomics. What else? I feel like well, this was very much an ad hoc kind of jumping on the streaming, and well, just start trying. But you can see that I did not prepare actually topics. Maybe for the next time. Next time it might be good to like prepare at least some search terms to check out. Or do something else, but still. Okay, genomics, let's say honestly speaking, any ideas from the chat? There is the chat thing on the should be on the right side of your screens. Happy to hear some ideas. Well, there was a that came to my mind that there was a movie with the medieval theme and with the apparent rabbit in it, but I think that was a bit different setting. Medieval rabbit. Well, that was a different. All right, so let's see. Let's go with the just classical, just to remain in the area of the same like archeogenomics, archeogenetics. I'm a bit skeptical about this. Well, this strange paper. It like before, it was also from this publisher, I think. But anyway, cattle remains, bones and teeth from both Roman and medieval age were collected in the archaeology. So this is the ancient DNA genomic amplification of Roman and the medieval bovine bones. Okay, so there's like bones and teeth. Roman medieval age were collected in the archaeological site of Ferento, Viterbo, Italy, in the aim of extracting and characterizing nucleic acids. Procedures to minimize contamination with modern DNA, ancient DNA kind of settings. Preservation of the archaeological remains were adopted. That's great. Different techniques to extract ancient DNA, like For extraction from bovine bones were tested to identify the method applies to peculiar characteristics of the study site. Currently, ADN investigation is mainly based on the mitochondrial DNA due to the ease of amplification of the small and high copied genome and to its usefulness in evolutionary studies. Okay, I didn't know this, but nice piece of information. So, well, I know that it's ancient DNA, but well, I didn't know that the empty DNA is preferred, at least for the ancient, so for the archaeological remains extractions, because of this reason. Well, it makes sense, but it's, well, I just didn't realize it. Okay, so preliminary amplification of both mitochondria and nuclear ancient DNA fragments from samples of Roman and medieval animals were performed, and partial specific sequences of mitochondrial D-loop as well as nuclear genes were obtained. The innovative amplification of okay, I'm just a bit, that is like they are confident, well, they are tapping themselves on the back a little bit, that they say that they are, yeah, but maybe they are. Innovative amplification of nuclear ancient DNA could enable the analysis of genes involved in the specific animal traits, giving insights of ancient genomic and cultural uses, as well as providing information on the origin of the modern lines of population. Nice, actually. If this, well, I don't know the details here, and honestly speaking, I don't want to go into details right now, but this one is, this last part is actually going into the same direction as the previous one, when they were kind of discussing with the Warhorses or Warhorses that they could figure out the color and the speed and the temperament of animals, and that is actually kind of, well, basically telling the same from a bit different aspect. This is for livestock, but it's actually basically the same, that analyzing ancient DNA by so basically sequencing and trying to figure out how they looked like. That's a very interesting goal in general, and like a very interesting endeavor. So it's a nice setting, honestly. Paris et al. Oh yeah. So Viterbo, I think they worked at home. Yeah, so it's but it's cool to have like the archaeological site at your working place. Université della Tuscia. Okay, good, nice. It's a nice paper as well. Medieval cataphone Bern. Genetical. Anybody from Switzerland in the chat? Animal domestication in the era of ancient genomics. What? Let's see. What do you tell us? Oh, it's a review. No, anyway. So, animal domestication in the era of ancient genomics. Oh, that's nice. Okay, or no, this is a different papers. But anyway, the domestication of animals led to a major shift in human subsistence patterns, from a hunter-gatherer to sedentary agricultural lifestyle, which ultimately resulted in the development of complex societies. Over the past 15,000 years, the phenotype and genotype of multiple animal species, such as dogs, pigs, sheep, goats, cattle, horses, have been substantially altered during their adaptation to the human niche. Recent methodological innovations, such as improved ancient DNA extraction methods and next-generation sequencing, have enabled the sequencing of whole ancient genomes. These genomes have helped construct the process in which animals entered into domestic relationships with humans ever subjected to novel selection pressures. Here we discuss and update key concepts in animal domestication in light of recent contributions from ancient genomics. Increase in the number of published ancient genomes for domesticated animal species and their wild relatives. Can I? Number of genomes. This is a bit unfortunate. Okay, like this. Number of what? More than one, less than 1x coverage. But come on, like, less than 1x coverage is not much. Oh, but this is an older paper. Oh, 2020. Oh, still. Well, what we see is a kind of market increase of well, in the last, well, until 2017 almost nothing. Well, a bit, but a substantial increase in the last years, and probably this kind of tendency just kind of continued in the last 5 years. Okay, and let's see the proportion. That would be kind of interesting. Horse, huh? Okay, it seems that the horse dominates. Like kind of, well, I'm looking at this one because it's like more than 1x coverage, but also here the horse dominates. Pigs, surprisingly. Okay, goats a lot. Horses and goats are the winners. Huh. Who would have thought? Cattle not so much, a little bit, but not so much. And the others, dogs are mentioned at least. That's interesting. Although, well, if there is a, I guess if they do like the archaeological sites on the where the people lived, and since the dogs, but actually all livestock kind of was living around the households at the time. So, but there is I mean, surprisingly, proportionally, I think there is quite a lot of dogs. I would not expect. And horses seem like, I don't know. Like quite prevalent, quite prevalent. That's interesting. Anyway, the influence of the ancient genomics on models of animal domestication. Oh, this is a way too complex figure for this time of the day. Well, no, I just skip this for now. Reconstructing population history in using ancient genomes. Okay, anybody interested in this part? So I see some people in the chat notably silent. Don't be afraid to say hi. Also, if you're not subscribed, actually there is a subscribe button, and your name then should pop up on the screen. I think there is an alert set in, so if anybody is not subscribed from the viewers, we might try out this feature and see if it works. Anyway, okay, so what we have here. Past decade, many tools have been developed, modified, tested for reconstruction of population history using ancient genomes. The first step in analyzing ancient genome data generated from next-generation sequencing platform is to align short reads to reference genome. Okay. The most used, well, this is again, I'm not very at home with the NGS data, but Burroughs Wheeler, that is kind of I guess, one of the more famous ones, which has been extensively tested with ancient DNA. Okay, assembling ancient genome instead of aligning to reference is often difficult given the highly fragmented nature of DNA molecule. Yes, actually, if you have seen and like because there are also open access data for ancient DNA, and also like SNP data, and also like NGS and everything. And if you open it, you would be amazed how fragmented the whole thing is. In terms of just fragmented means a lot of missing, like a huge portions and chunks of the genome are missing, for example in SNP data and something like that. So that I can believe that. So this is kind of, if they are kind of referring to the fragmented nature of ancient DNA molecules, what they are saying here, this is that they are just a whole lot of parts of DNA are missing. So it makes this the whole archeogenomics kind of analysis that much more challenging. Oh, actually, they are telling the same, making the ancient genomes fairly difficult to use when no reference genome is available from the closely related species. Well, it's kind of but see references. Okay, there is some solutions for that. Following short read alignment, the genotype of an ancient sample can be inferred by comparing the sequence of short reads with the reference genome. Single nucleotide polymorphisms are favorites, or mine. SNPs, which are the result of single point mutations, are the variant type most commonly used to reconstruct population history. Yes, because it's very well adapted. Well, just kind of the SNPs are quite nice variants to do that, because there are many of them, and you can just compare quite a bit the individuals among themselves, and if you're lucky also you can compare the individuals to kind of historical individuals to today's samples. Other types of structural variation, such as copy number variation and insertion or deletion, can also be inferred, although working with this type of variation can be challenging owing to the issues inherent to the ancient DNA, for example, small molecules. Okay, but nice. The nice thing with the SNPs is that it's just a single point mutation. So there is, if that part of the DNA is there, then all you need is one point or one nucleotide that is changing, and you have already a SNP. Whereas for the others, for example, insertions, deletions, you have to have kind of uninterrupted segments and have a luck to have the kind of the variant, the insertion or deletion within that uninterrupted sequence. So yeah, I would guess this would be a very valid statement here. And again, this is something that I did not consider before, or well, I was just not thinking about it, but just kind of because I knew about this fragmented nature of the nucleotides, but or the samples, but not too much about I just didn't think about it in connection of the insertions and deletions. The genotype of ancient genome is then compared with those of other ancient and modern genomes. One popular approach is to compare genotypes as SNPs that have been pre-ascertained in the modern population. Okay, this method minimizes the incorporation of enormous SNPs arising from ancient DNA damage, such as deamination, in downstream analysis, although it can also introduce biases. Many population genomics tools exist to infer population structure by comparing genotypes as SNPs on multiple ancient and modern samples, some of which have been tailor-made for ancient DNA. Principal component analysis is one of the most commonly used tools to infer ancestry. Okay, it's not very complex, but okay, why not? And accommodate highly degraded and low coverage sequence data by allowing an ancient genome to be projected on the principal component axis. Okay, so this is some kind of imputation then. And the other thing that I was thinking that why not use that, but they are continuing with that train of thought. Other tools such as the model-based clustering method admixture can also provide information about the population structure and admixture in set of modern and ancient samples. This is what I was, well, I was not reading ahead, but when they were talking about, here where it was, yeah, so inferring population structure and stuff like that, it came to my mind that why not some kind of admixture result. I mean, because that's admixture is nothing else than just showing the similarity between the individuals and well, basically determining if they belong to the same population or not, or if there is a part of a population or an individual is a composite of multiple populations. So that is basically what admixture is doing in a very simplified description. So, and apparently it works also in the Archeogenomics samples, which is good news. Then basically what you can do, which might be an interesting thing to test out, is to download a bunch of these open access archaeogenomic samples and run through with admixture analysis with other open access current samples. Well, and see what comes out, if there is any connection. Yeah. And of course, if you can design the study in a better way, that it's you have specific genotypes, either archeo samples or current genotypes, that you have a like kind of a plan to compare things, then of course it's a much more targeted approach. So what I was telling, this downloading open access is kind of very much like shot in a dark, or how to say, like kind of a, yeah, well, see what happens approach. But might be viable. That depends very much on luck, actually. But yeah, why not? Just the problem is that there are too many things to try out, or possibilities to try out, and there is too little time. Anyway, so additional methods based on asymmetry in gene trees, D statistics, or allelic frequency correlations, F statistics, FSTs, yeah, so we deal with FSTs with actually also on the channel. So if you're interested, actually it's one of the easier things to do, the computing. Well, F statistic, of course, is much wider than that, but FSTs are part of that, and I guess here it's there also, because the FSTs are basically about the similarities of two populations. So this is very much along the lines of admixture, and here I did this. I think they must refer to FSTs, and actually you can do basically the same things. So they download ancient samples, download modern samples, do admixture, do PCA, and do FSTs, as well as more explicit graph testing approaches. 3-mix admixture graphs are robust tools for testing existence of gene flow between populations. Nice. Actually, that's maybe not a bad idea in general, just to check on also in the 3-mix and other stuff, also in the Genomics Bootcamp channel in general, because yeah, these are valid approaches, of course, to today's populations as well. Okay, so we you see it's a very long paper, so we just fast forward to the end. But kind of well, I didn't want it to read this one, it's just but that this box was far too interesting to skip. It seems that the reconstruction of population history using ancient genomes, but now we can go through methods in ancient genomics, early domestication, the origin of domesticated animals. They are dealing quite a bit on that. Genetic integrity during domestication, reproductive isolation, founder effect, later stages of domestication. So they are dealing with movements of various species, some of which is questionable, or not questionable, but it's like not exactly proven, it seems. Spatio-temporal pattern of admixture between cattle and zebu. Divergence of European and African cattle. This paper seems to be quite a ride. Animal husbandry, a lot of topics touched. Artificial selection by time. Some of our favorite genes here, MC1R and similar. Applications, conservation, animal health, perspectives. Well, this is one of the longer papers, to be honest, and the references. There is no conclusions as such, so I will leave it here. But yeah, definitely. Perhaps if you're interested in general, in domestication, or archeogenetics, or like generally like how to connect to genomics to more ancient samples. So this paper from Franz et al., Animal Domestication in the Era of Ancient Genomics, is definitely something to check out. So good. So and yeah, so in the description I wrote that I would have a 1 hour stream, which we are a bit over that, but anyway, so I want to thank to everyone who has been here. Well, I was kind of thinking to if honestly speaking, it's a bit different kind of setting that I usually do in a Genomics Bootcamp channel. But I think for the time being, I will leave this video on the in the live stream section of the channel. And anyway, so you can check it out. And well, let's see if you are interested in a similar content with papers or with anything else in terms of live streaming on this channel, then could be a Q&A or anything, but it's like in this format of the live streaming here and there. So then for sure let me know, and yeah, and then leave a comment in the comment section below for this video. Anyway, so for now I would end it here. I definitely want to thank all of you who were here. So I saw that there were people kind of popping up, although well, silent in the chat, but I saw that there were people kind of popping up, which I'm happy about, even though this was absolutely unannounced stream, just very spontaneous. But anyway, so what I want to say is that let me know if you liked it, and then we can do more as well. For now, for sure, I thank you for your time. I hope you had a nice day. And if you are kind of a same time zone as me, and you have like 00:15 a.m., then I also wish you a good night. So that was it from my side. I thank you very much, and well, I see you then next time. Have a nice evening and have a nice day also for tomorrow. So bye then, and